So we're in your chemistry in chapter 18, section 5, and this chapter section is on biochemistry. In fact, it's the last section of your book, and so we'll be done with chemistry for the year. So biochemistry, the study of chemistry of living things. Living cells were once thought in history, before the 1900s and 1800s, they thought living cells were just simple little bags of organic chemicals that were energized by some vital force. But at every turn now, we know the study of biochemistry reveals the hand of a creator that loves us and a creator that is very intelligent, because in the midst of biochemistry, we have these tiny, tiny cells that are basically not so simple as they thought. They're not a simple goo, but scientists have found microscopic pumps, motors, sensors, turbo generators, information storage and retrieval systems, complex mechanisms and highways in the basic cell units of living things. So when you think of what biochemistry is about, you think how amazing creator we have. You can see these pictures here. We have some pictures of the alanine, cysteine, asparic acid, serine, glutamine, histidine, phenylalanine, and lysine. You see how complex and how very different each one of these cells are organized. You know, these molecules are organized. And you can see in the midst that it just gets to be a very complex system, not like they thought in the past. So let's start with proteins and amino acids. So as we can see, proteins are made of amino acids, but proteins and amino acids, I should say in itself, represent 50% of the cell's dry weight, 50%. So very, as for amino acids in the cells, very complex compared to other non-living things, right? So we have organic polymers, and these organic polymers make up and that maintain living cells. So, and the polymers basically are presented with four different elements: carbon, hydrogen, oxygen, and nitrogen. Now we're going to get some phosphorus and such in there, but the basic structure is going to be of these four elements and how they're organized in proteins: carbon, hydrogen, oxygen, and nitrogen. And there's going to be a variety of chains, but the amino acids usually come in about 20 different amino acids. So about 20 different kinds. And you can see how they're organized here. So we can look, you'll see an amine group. We know this is an amine group because it has an N. N means amine group. And then it's going to be united by a peptide here onto the other part of the group, which would be the carboxylic acid group, the carboxylic acid group. So we have the on one side, the carboxylic, and the other side, the amine group. And this is what makes up all proteins. Just remember, there's two different sides, amine and carboxylic. The carboxylic is known for the acid, carboxylic acid, right? So going on, we also have what's called peptide bonds that bring together each one. So each you have a different amines, they're held together by what's called a peptide bond. And so here we have one protein amino acid. Usually there's like all of these different amino acids coming together, you know, so we have in between these two amino acids, number 1, 2, and going on, the different kinds of amino acids, you'll have a bond. And see how these two are coming together. Here shows a picture here. They're bond together here with this peptide bond from the carboxylic onto the amino, and you'll see where they're bonded together there. So to know, it's not that really complicated, but in a sense it is, because you can see how these form together, and forming into what they are, into a living cell, is pretty amazing. So let's go on to the next section here. Whoops, here we go. So the protein structure and the protein function. So we have a different structure of the proteins. Basically, there's the fibrous and the globular. Fibrous and the globular. Here's a picture of the fibrous protein, which is collagen. You know, you always think some people take collagen, you know, drinks and pills and stuff to help with their skin and their hair and such, because it's a fibrous protein. It's fairly straight, and it provides structure to the cells. And it's 1/3 of the proteins in the body actually are collagen. The structure not only in like I said, in your hair and skin and cartilage, it's also in your bones. So this is the fibrous protein. It looks very strong, as you can see here, and very straight. And on to the globular. Here's the globular protein. And there's quite a few different globular amino acids in your body. Some of them are in like hemoglobin, you know, so it sounds like hemoglobin, right? So hemoglobin is one. That's in your blood, your hemoglobin in your blood. And it transports oxygen. And there's insulin, is a hormone that helps regulate blood sugar. So you have these different, also different enzymes. Different enzymes are also globular proteins. So we have the globular protein, and this is a picture of an insulin protein that's in insulin is an enzyme which helps with your blood sugar, right? And here's the fibrous, those two different kinds of amino acids in our body. This little article is really pretty amazing. So as you see here, it's going to talk about really how complex our cells are. And when you're going through it, you think, oh, how could this just happen? Well, it couldn't. You know, God has a plan, even in the midst of each little building block of our body, each cell. So molecular motors, they're going to talk about, there's little motors in our cells. So there's two different kinds. They have the dynein, and they have the kinesin and dynein. Those are the two different types. You know, cells, basically these motor proteins, they're called motor proteins, are cells to reorganize our internal components and to, you know, they basically, so that our cells will work in the midst of them. And also they can change shape, and they move from place to place. They transport materials and equipment in our cells place to place. They actually operate on tracks. Can you believe that? Like little networks of railroad trains or something inside of us. And they ship things from one place to the next. When you think of FedEx or something, you think, okay, we have these in our cells. They basically attach to several. There's the vesicle that attached to several of the kinesin and dynein. These on these microtubes, and these little motors powered by ATP. ATP is the energy breakdown that happens in the cell that gives these little motors enough energy, enough fuel to travel in. So they even travel in opposite directions, like a 2-lane highway. You know, so here's a picture here. These dynein motors here. This is the membrane. This is the microtube here onto the different proteins and the different trucks and stuff in the midst. These are in protein. Also, they present like flagella. If you take a simple microorganism, or I should say organism, like a bacteria, they have these flagella that sweep, or cilia that act like oars in our body, even, you know, to propel water through the cell and such. These little celiac act like sweeping, like to sweep the water, sweep the food and such across. These are made of these fibers too. Myosin, basically, a myosin is the basis muscle cell that helps the cell contract. This also is powered. Here is myosin, the basic muscle cell, and it's actually made of these fibers and powered the same way. It's like the muscles are reeling in the cables, shortening the cell. As they do this contracts and expands our muscles, you know. So let's go on to the next. Hey, this picture is pretty amazing here. So it says here, the flagella of other organisms, instead of being a bendable tail, the bacterial flagellum is more like a spiral propeller. And to move the bacteria spins the flagellum at several thousand revolutions per minute with a high-speed rotary motor. So this inside our cells, we have these high-speed motors going on. Here's a picture of it. Here's a picture of the flagella. Here's the reversal, the mechanism that's in the cell, forward and reverse. They reverse. They can basically, you know, change directions. Here's the cell wall. Here we see how the flagella swims here in the midst. Here is a small picture of the bacteria here with the flagella, and onto here you see them where the motor, the control proteins are. It says CYP or motor A, you know, stator motor A. There's names for them. So this is the bacteria drive motor, and there's also similar things in each one of our cells. So it also is powered by electricity. When you say electricity, it's the electricity, electrolytes can power these, and powered by electrical charges of the protons and the hydrogen ions. So, and amidst it even is run by electric. So this is just showing to our careful design that God has put his precision on his engineering, basically precise. Living cells could not have formed by mere chance. This is a testament of an intelligent and skilled creator. So I love this picture. Can you imagine someone that says, okay, these things just happened. There's no way that this could happen. You have the motor engine of your car, and we say, no one built that. It just happened, it evolved. There's no way. So how amazing are our cells. So we talked about the protein. Now let's talk a little bit about carbohydrates. Carbohydrates too are another important molecules in our body. So carbohydrates are the most important energy source in compounds, and they are mainly from sugars, and they are organic carbohydrates. They belong to the group of organic carbohydrates. Carbohydrates are aldehydes and ketones. Okay, remember that. They're aldehydes and ketones. Aldehydes and ketones that contain multiple hydroxyl alcohol groups and compounds derived from such aldehydes and ketones. So sugars, we divide these sugars into two groups, monosaccharides and disaccharides. And so what's the difference? Well, we'll see mono meaning one and di meaning two. Two molecules on the disaccharide, and the mono only has one. So the monosaccharide is the simplest form. It's usually 3 to 8 carbon atom molecules, or some actually going mainly concentrating on 5 or 6. It's the common monosaccharide. There's pentoses, hexoses, and they're the more stable because they're in a cyclic form. So you see here's glucose is kind of an open chain here of sugars. Here's a glucose ring forming here, and here's a fructose open chain. These are some of the molecular, structures of how they're structured together with glucose. And disaccharides two molecules of monosaccharides form together to bond together, and they make more complex sugars like sucrose, which is table sugar, lactose, which is milk sugar, and maltose, which is malt sugar. So here we have some rings that were formed. Here's another with these are all monosaccharides right here. They're all different. This is glucose ring form, glucose open chain, fructose, galactose ring, another mono, onto ribose ring, another type of. This is kind of a shaped with the five-sided, penta formed pentasaccharide, I should say, instead of mono or di. So pentosis and hexosis, they call them actually. So let's go on to the next here. So as for disaccharides, there's another kind of saccharide called a polysaccharide. So let's find out what a polysaccharide is. Oh, before we get to the polysaccharides, look at this picture here of the disaccharides, and you can see how they're put together. These two, this is a glucose and a fructose coming together, and you see how they're united here by the oxygen, ring here. So here we come, as they come together, you can see now we have glucose and fructose coming together, making sucrose, and then water, sucrose and water. So here we just have, that's how the disaccharides come together. So onto the polysaccharides, and you can see some pictures here. Here's a polysaccharide. You see now, not just two are coming together, but we have two or more coming together. And so we have some kinds of polysaccharides. Amylose. Amylose is a common, and it's an unbranched polymer of glucose. We have amylopectin, and it's amylose, but it has branches along the chain. We have starch. I'm sure you've heard of starch in food. And starch is usually a short-term glucose that's manufactured in plants, and it's a mixture of amylose and amylopectin together. And then we have, that's in animals, in man, is glycogen. And glycogen is animal starch, so, and it's, glycogen is used for storing glucose. It's similar to amylopectin, but has more branches, it says. So God has designed the cell to condense millions of glucose molecules into just a handful of these polymer molecules. And this keeps the cell from bursting under its own osmotic pressure. So because God's design is so specific, if it would be a little more, the osmotic pressure could not hold it and the cells would burst. So it's just perfect, into holding these millions of glucose molecules in this just a few polymers. So you can imagine. So here's a picture, polysaccharide. And we have, again, amylose, amylopectin, starch, and see how they're organized, 1, 2, 3, 4, and how they're put together right here with the oxygen binding here. So carbohydrates. Cellulose, a little bit more about cellulose. So cellulose is another type of polysaccharide, and cellulose is in wood and cotton and plants, and it has unbranched polymers of glucose. It alternates in glucose manometers, so it's flipped. It actually is flipped a little opposite than the amylose. But anyway, cellulose is much stronger because of it. So here we have glucose alternate monomers flipped till we have cellulose. So here's a picture of cellulose. So and it's much stronger in the midst of that. So what about this cellulose makes it stronger than starch, and it's also insoluble in water. See, humans and animals do not possess enzymes to digest cellulose. So when we eat cellulose, it's actually good for us because it actually is good for our digestion, to keep our digesting going in our intestines. And it harbors intestinal bacteria that actually produce enzymes, cellulase, and to digest the cellulose inside of it. So we have bacteria that work on our cellulose, the cellulose inside of us, and also a lot of it pass through and is used to help in digestion. So cellulose. But plants make cellulose. That's where we have cellulose in wood and cotton and plant. All kinds of plants have cellulose. Okay, let's go to the third section. We went over proteins, we went over carbohydrates. Now let's go over lipids. Lipids are what? Lipids are fat. When you think of lipids, you think of the fats in our body. Lipids are a little different. Let me see. Lipids are a little different because they're characterized, by their insolubility in water, because they're like oil-like with the fat. And the body uses lipids for a variety of functions, including storing energy, building cell parts, and transferring chemical messages between cells. So lipids are very important. So you need to have enough fat in your diet, in the midst of having lipids and bringing forth the lipids in your diet. Let's see here. Okay, fatty acids and fats and lipids. So fatty acid is the simplest lipid. That's a question we need to know. A fatty acid, it has a carboxyl acid, carboxyl acid that's branched on a chain of 12 to 24 combination of, the fatty acid. The carboxyl acid onto this, and it can react with alcohols even to form esters. So you have, actually, when I say variety, there's 3 fatty acids that usually react in the trihydroalcohol system, and they form, the 3 fatty acids form glycerin. So they're called triglycerins. So if you see that any place, triglycerin. So let's look at this picture here. Here's the simplest type of lipid, and it's a carboxylic acid with unbranched chain here. So we have one end here has this carboxyl acid at one end, and then they have the chain goes on, as you see. And so here's long. See how long it goes on and on. This is a fatty acid. This is stearic acid right here. So it has a chain of 12 to 24 carbon atoms. Fatty acids can react to, like I said, to alcohols to form esters. And so if 3 fatty acids react with trihydroxyl alcohol, they'll become glycerin, which are glycerol, and this is that triglyceride or fat. Like I said before, kind of repeating myself here, but here's a picture of a fat triglyceride right here, with all the different going through very polyorganized here, right? So you have polymer. These are all polymers to the end, and at the end you get the glycerol portion here at the end. So things, what are these fatty? Here's another picture here of glycerol, how they're hooked together with 3. It says 3 fatty acids react together and hook together with 3. That was 1. Now we have 3 of them here. And this becomes the glycerol portion here again, and the fatty acid portion put together. So we have, they are used in things like, are basically made into things such as shortening, vegetable oil, lard, and animal fat are all mixtures of these triglycerides. Fats are used by both animals and plants to store energy, provide more than twice as much energy per gram than carbohydrates. So a fat does give you more energy, although fat is stored a lot, and a lot of times we have excess fat in our body, don't we? And fats that are liquid at room temperature are called oils. We know that. Now there's 2 different types of fatty acids, and you've probably heard this before, but we'll go over it again. The 2 different types are the saturated fatty acids and the unsaturated fatty acids. And so those two are important, and the saturated are stack easily together, causing them to exist in a solid form at room temperature. So saturated are at room temperature, they're solid. They're a solid form. But fatty acids that are unsaturated, they do not stick well, and they have these double bonds, kink chains they call them, and these fats are usually at room temperature. So like if you have olive oil, that would be unsaturated, and then you have a stick of butter, that would be saturated fat. So two different kinds of fats. It says, since triglycerides are esters, they may be hydrolyzed to form fatty acids and the alcohol glycerol. If the reaction is performed in a basic medium, the salt of the fatty acid is obtained instead of the acid. So basic hydrolysis of a fat or oil is called, this is a word, saponification. Saponification is the hydrolysis of a fat or oil. The fatty acid salts then are soaps. So when you think of saponification, I always think of soaps then, right? So basically it's the hydrolysis of a fat or oil. So here's example of that reaction here. So we have basically here tristerion. We have a glycerol portion and the fatty acid portion there of our fatty acid and of our molecule there, and then you can see how the glycerol becomes sodium stearate, which is soap. As it divides out, the molecules change in order, and we have glycerol and sodium, and we have a soap. And it says softer soaps are obtained by forming the potassium salt instead of the sodium salt. So just saponification is producing soaps. So you may need to know that word. So here's the structure of a soap molecule. See how complex it is here too? Here's the polar handle of one. This is going to be, the handle here would be the glycerol portion at the end here. And this would be polar. When it looks like this, it's polar. Looks like this, it's non-polar. So in that way, it would structure out to be a soap molecule because they have a polar. Usually soap molecules have a polar side and a non-polar. Remember, water is polar. So when you put this polar handle in, it basically is likes, dissolve and like. So it's the portion that would dissolve, and then the other portion is not. And this way it dissolves and takes out. That's what soap does. Dissolves and takes out the dirt. Phospholipids. So phospholipids are another. Here's some pictures of phospholipids. Phospholipids are important components of the cell membrane. Just remember that. We need phosphorus to be in our cell membrane so that we have that our cells working properly. So here's a picture of the phosphorus here. This is the phosphate head here, and then we have the fatty acid tail. Typical phospholipid. This is lecithin, which is a phospholipid. Here's the structure above here, how they look with the as for the structure, and this is what you would think as the molecules come together. So, and instead, having that phosphate group. This is phosphate. It's phosphate group at the end here. That phosphate group. In water, phospholipid molecules form two-layer membranes with the polar phosphate heads facing the surface of the membrane and the tails on the opposite. So, and that's how they form. But here's the picture of the way they form. So each one of these is a phospholipid, and as it's coming together, it will form the cell membrane of our cells. So pretty cool. Another are the steroids, and steroids are a group of lipids characterized by a structure consisting of 4 rings. So now to know what kind of steroids, what are steroids in our body, because you hear the word steroids a lot. Well, steroids, of course, we're talking about what? We're talking about lipids and fatty acids. Well, steroids consist of these four rings and organized in ways, and you'll think of some of the things in our body, such as cholesterol, which is the most important steroid. It manufactures all of the other steroids come from cholesterol, and there's a component of the cell too. So cholesterol is very important, and other steroids include bile salts and vitamin D, both, and all hormones are steroids too. So here's a picture of steroids. Here we have the steroid ring structure. See how it's put together? A steroids kind of here, and you see how the ring here, and here again we have a steroid. Cholesterol being the major steroid. Probably a test question like, what is cholesterol? And you'll say, oh, it's a steroid. What is steroid? Steroid is a fatty acid. So I'm going to go on and finish this up. So we have, the last section is on nucleic acids. Nucleic acids are very special because they represent the biomolecules in our body that guide and direct the construction of all other proteins in cells. So these are where our plans, the plans God put inside of our cells and what they're going to be. It's our heredity. So our heredity is so important. It passes the traits from parents to offspring. And there are two main types of nucleic acid. One are DNA, deoxyribonucleic acid, and then we have also RNA. So DNA, ribonucleic is RNA, and deoxyribonucleic is DNA. And so DNA basically gives us our what we look like and what our cells do. It holds the heredity of the next cell, what the next cell, and what the next genotypes, genes are going to be. And these are called nucleic acids. DNA and RNA. And let's talk about nucleotides. So nucleotides are acids or polymers of molecules called nucleotides. So nucleic acids are nucleotides. So nucleotides, each nucleotide consists of three parts: a nitrogen containing base, a sugar molecule, and a phosphate group. In the RNA, the sugar is the pentose ribose, but in DNA, the sugar is deoxyribose. So that's why it's called deoxyribose. DNA, deoxyribose, is a type of sugar that's inside of here. And ribose is in the RNA. So, and then it's formed by removing the hydroxyl group from the ribose. Anyway, when the nucleotides polymerize, the sugar and the phosphate groups alternate to form the backbone of the nucleic acid. Nucleic acid, just think of DNA and RNA. So basically, knowing the nucleotides, these nucleotides have a nitrogen base, sugar molecules, and a phosphate group. So let's go on here. The structure of the DNA. The structure of the DNA, we know that the DNA is a polymer, right? That means it goes in long strands, chains. It's a single backbone, and there's a double backbone. The RNA has a single backbone, and the DNA is double. They call it the double helix. The double helix is DNA. So that's the RNA has several structural differences, and you see it's the type of sugar that it is, and it also is basically, as we see here is the form. Of course, single would be RNA. It's just a single, but here we have the double helix, and this is DNA. See how it twists on itself like a ladder, like a spiral that goes up. And here's the bases. The DNA structure bases, they come in 4 different types of proteins, nucleotides. So nucleotides thymine, adenine, cytosine, and guanine. So these will come 1, 2, 3, 4, and they'll match up into the pairs, and the pairs will be in between. The pairs match between one side to the other side. These pairs matching up on the ladder. So this is the basic structure of the DNA. So, and guanine always, guanine always pairs up with cysteine. These two always pair up together. And adenine always pairs up to thymine. So let's go to the RNA structure in the next here, so we can have that too. Oh, well, anyway, I thought we had a picture here, but we don't. The RNA structure is a little different. The RNA structure, instead of having the thymine, this would be gone. This type of nucleotide would be gone, and it would be called uracil. So with the RNA, it's going to be a uracil instead of thymine. And the RNA is going to be just a single strand instead of a double strand. And the RNA, the sugar is a ribose, and not deoxyribose, is ribose with the sugar. And it folds, RNA kind of folds in on itself because it's just a single instead. RNAs are the messenger, they're called messenger RNAs, most of them, that gather messages from the DNA to the ribosome, so the ribosome can produce another cell. So it's very complex, but you can imagine how would you ever think that this could happen on its own? We have a wonderful creator, don't we? So it says information in the nucleic acid, the sequence of nitrogenous bases in DNA encodes the information of the cell. So, and God has created complex cellular mechanisms that allow the cell to use this information. Various proteins work together to make RNA copies of the DNA information and to transfer these copies to protein factories called ribosomes within the cell. The ribosomes read the information to determine the correct amino acid sequence for each protein. This information system within the cell typifies the amazing precision and design that characterizes the work of all wise, the all-powerful, the all-wonderful Creator. So let's go ahead and stop there. And we're actually finished with your book, and I'm going to go over some of your quizzes now. We went over, I'm not going to go over these questions because I went over everything that you can imagine. There's two types of nucleic acids, RNA and DNA. When you look at this application, you think, what does this present? And that basically that whole presents. We're talking about what? A DNA and RNA. How complicated it could be. So I pretend it actually represents a nucleotide. So, okay, let's go ahead and stop there.